From MicrobeTV, this is Immune, episode 107, recorded on July 8th, 2026. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, it's finally summer. Are you warm?
It's almost over. It's July. Look
Here
how fast
it's just starting.
Look how fast June went. Actually,
I know.
it was really hot for a few days. It was over 100.
Very
now,
hot.
Then it got cool the last few days, and then finally it's like 80 something, and
It's been
pretty
spectacular
nice
here.
today.
Yep.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is also pretty spectacular out today after going through the same overly hot and overly rainy and windy and stormy.
And humid.
times.
Very humid.
Yes. We had some pretty bad storms here.
Yeah, a lot of trees down, right?
Mm-hmm.
So one guy was driving, a tree fell on his car and it killed him.
Oh,
Just right down the street
no.
from my house. Yeah. Poor guy.
Were your trains running?
Yes,
the train was shut down because of trees on the track.
Yeah, here they were running. But that often happens. You get a tree on the track, forget it. Yep. Also joining us from Cleveland, Ohio, Steph Langle.
Hi, everyone. Good to see you. Happy summer.
Happy summer.
Yeah, things are good in Cleveland. We've had storms. It's been very hot. It's a little bit better now. So are you guys traveling to any other conferences in the next couple months, or is that kind of at bay?
ASV is coming up.
Oh, yes.
Yep.
Yes, I can't go this year. But it's in Minneapolis.
Mm-hmm.
Yep. So it turns out there is a measles mini symposium that weekend before ASV.
Oh.
And so I'm going to go
What is that
to
hosted by? A different group, or ASV is
No, it's
hosting?
hosted by Catania and somebody else there at the Mayo Clinic. Yeah.
Oh.
And
nice.
he asked me to go and maybe do a podcast. So I'm going to go
Oh, that would be
see
nice.
that. Brian, I was just in Fort Collins a couple of weeks ago.
Oh, fun.
That was fun. I enjoyed that very much. I'm going to ASV, and then I'm trying to arrange a trip to Finland in August.
Oh, for what?
To go to, there's a city called Turku, and there's a university there with a lot of virologists, and go there and do a podcast.
Did you get to see the bats in Fort Collins?
I did the second time, yes. So the first time I saw them, I walked actually in the cage and they were flying around. And this time they were all separated. But yes, I saw them. They have a new facility where they put them. It's really beautiful. Yeah. See, they told me the biggest expense is the bat food.
Really?
They
What
say
do they eat?
all kinds of fruit. He said they have a semi pull-up every week full of fruit. And a lot. They eat a lot of fruit because they're fruit bats.
Yep.
But it was nice. It was very cool.
I guess
And,
it might be even more expensive if they were insectivorous and you had to always
Get
maintain a
insects.
colony of insects, huh?
Yep.
The bats in
would
my
be
backyard
something
eat the
else.
insects. They're always flying around at dusk. It's pretty cool.
If you enjoy these programs where we talk about science, real science, not lies that you hear every day. Oh my gosh, the lies these days. Anyway, we will not get into that, but we would love your support because we don't put ads on these programs. I don't really like ads. They're disruptive. So go to microbe.tv/contribute. And you don't have to give a huge amount, although if you'd like to, that would be great. I'm not saying it to rule it out, but you could just give us a cup of coffee a month to help produce these programs. There's a lot of them. There's like 10 at microbe.tv.
Yep.
And just to show you why you should support us today, we have two amazing papers. And I'm happy I just ate lunch for the first one, which Brianne is going to tell us about.
Yeah. So I came across this paper because I get Nature papers coming to my inbox, and I actually saw the name of the podcast that Nature did talking about it, which was called Immunity Gets a Boost from a Surprising Place Breakfast. And I clicked immediately. Largely because I'm always kind of interested in the kind of ways that people talk in general about what you need to do for your immune system that maybe have been going on for a long time, and whether there's actually data behind that, whether the fact that, eating your breakfast is, something that, is great for health, but is there actually data behind any of those types of things? So I usually click on papers that get at areas like that. This paper,
Brienne, should I have chicken soup for a cold?
Right. That's a good question.
And Brienne, you had said your class, your students ask a lot of these questions. And I will say when I share what I do, I get a lot of those type of questions about what should I eat? I'm like, I study not that, but
same thing. At the beginning of the semester, every semester, I have a little survey, and I always ask the students what types of things they're most interested in learning about. And the number of them that want to know things like what they should do to help their immune health and what they should eat and things like that is pretty high.
I think it has a lot to do with them seeing things on social media that
I do too.
Because there's a lot of, as Vincent was saying, misinformation that's out there. But those are things all the time. And those people speak so decisively and convincingly. You must eat this, you must not eat that, and whatever. And people who are, I guess, inquisitive are like, is that really true? Mm-hmm. So then they are like, maybe I'll take an immunology class and figure out if it's true. And then we tell them, well, we have no idea. And they're like, wait, now I'm back to square one.
But even this is not eating, but so people write to us all the time. I took a shower, went outside with wet hair, and I got a cold. People are convinced that did it, right? And there's no research that addresses that.
Yeah.
There is a paper.
Oh, is there?
Well, let's do it.
We should do it. I think the other hard thing is a lot of it is it's kind of presented as here's a hack or here's what they're not telling you. And I think there's something about human nature. People are just like, oh, what are they not telling me? But when I talk to family physicians, like family medicine, they're like, eat vegetables, whole grains. But that is difficult for people to do. So I think it's the things that are so clearly associated with health are hard for us to do. And I think the selling pitch that people do is it's just not right.
I hear nutritionists say you eat lots of vegetables, get a lot of vegetables, whole grains and proteins, right? Mm-hmm. And it's boring. And that's what
just boring.
Don't eat sugars, don't eat these other things. But yet most people, like you said, don't even meet those minimum requirements. And sometimes it's not through their own fault, right?
For sure.
Because there's a lot of food deserts and things. People can't even get fresh fruit. But anyway, so it's just do the basics and your immune system should be fine. But there's no trick or hack.
Right. Which, Brian, I know this is a little bit of a digress from this paper because this paper is telling us kind of specific things, but just to speak to, I think the intersection of immunology and nutrition is fascinating and really we need to do more.
Yeah, absolutely. Yeah. And I think that throughout this paper, I could imagine, other questions or other things that they could that one could examine as sort of next steps. And I actually even really liked in the intro, they kind of talked about the big picture of the immune system and metabolism. And, I was even more impressed by the paper after I'd read the intro because the intro had convinced me that I didn't think that this experiment would work. So
they were good at storytelling.
Yes, absolutely.
They got you hooked.
Absolutely. So they point out that we know that there are important ways that T cells are controlled by different metabolic states. T cells will do different types of metabolism as memory cells or naive cells. And they sort of talk about the fact that there's a lot that's known about that. They talk about the fact that there's a lot that's known about kind of long-term diet manipulation and immune responses. And they mention all of those things. But then they also mention, which is that I hadn't really thought about, that, T cells have evolved to, circulate around the body to lots of different tissues with lots of different metabolic states and lots of different demands for energy and things like that. And so that made me think, so then I guess short-term things probably wouldn't matter as much because the T cells are circulating. They're going to be in such different metabolic conditions that maybe a shorter-term type of thing won't work, unlike some of the longer-term things. And so in this paper, which I forgot to give you the title, it's called Postprandial Lipid Metabolism Durably Enhances T-Cell Immunity. The first author is Alok Kumar, and the senior author is Greg Delgoff, and they're all from the University of Pittsburgh, at different departments of the University of Pittsburgh. Here, they started looking at T-cell responses, specifically CD8 T-cell responses for most of the paper, in individuals either after an overnight fast or 6 hours after eating. So basically, they have a group, at least to start with, of 31 patients who fasted overnight. They took blood after their overnight fast, and then the individuals had a meal, and then 6 hours after the meal, they took blood again. And they looked at T cells, both after the fast and then 6 hours after the meal. When they looked, they didn't see any big changes in T cell sort of markers, but the T cells had big differences in metabolic capacity, measured with a lot of different types of metabolic assays, looking at things like glucose uptake, mitochondrial mass, oxidative metabolism. And so after eating, the T cells seem to have sort of increased metabolism. They also noticed that the T cells postprandial or after the meal had higher effector functions. Specifically, they were making more interferon gamma and TNF alpha. So it seemed like the T cells had better effector function. And in fact, if they took those T cells out and put them in a culture, the fed T cells, as they call them, maintained advantages for 7 days in culture. And so it seemed as though those fed T cells were acting better. They seemed to have a higher proportion going into central memory. And they did some really nice controls to show that this wasn't about sort of circadian rhythm. It wasn't about just the timing of when T cells were drawn or things like that. It was actually about the fasting.
I have a question.
Yeah. Okay. And I also have a funny side piece.
I have a thought and a question that I might have just forgot the question. But the thought is that, doesn't this make a little bit of sense to me? Because when you eat is when you have a significant opportunity to be exposed to potential pathogens via fecal oral route, right?
Right. No, very true.
So I think it makes sense. But I'll say my question is, isn't it a little odd that those T cells maintain that state for 7 days? Because that would suggest that once we eat once, our T cells are always on. So how are they measuring them as off?
Right. So they're basically just measuring in this experiment. They do some future other experiments to address some of these same kind of things. Here, they're basically just looking at the ability of those T cells to produce cytokine and to expand in a dish. And so it's basically, do they have that enhanced ability or not? I don't know that they're actually necessarily saying off, but it's more of a, are they enhanced or not enhanced type of thing.
But I mean, the mouse has been eating its whole life if they collect the cells. So.
Yeah. So they, so I haven't gone to the mouse stuff yet.
Oh,
but right here, they're looking at people first.
Got it.
But you're right. Like in either case, they're looking at
like 6 hours or 8 hours ago, 12 hours ago.
They, later, when they get into the mouse studies, they're able to look at antigen specific versus naive cells to see if it's about, did you see the antigen during that time after feeding, or was it a naive cell? And I think that because of what they talk about with some of the serum lipids, it's sort of about what kind of serum lipids are around and what kind of signaling is happening as the T cells activated. That becomes really key. But I think all of these questions are really great questions. They also have a table that's a supplemental table that I spent perhaps too much time on, but I laughed a lot, which lists the 31 participants and the meal ate the last thing they ate before the fast, and then what the meal was, and then what the meal was that they ate after the fast.
Oh.
The last supper.
Because it wasn't standardized, correct?
No, it wasn't
they didn't
standardized.
Make everyone eat the same thing.
No, and so it's really varied. I don't think the sample size is large enough to really make any conclusions, but I don't see anything about specific foods being different. But I did want to commend, the people for their honesty. Like the person who's 6 hours after the fast meal was chocolate candies, coffee, 3 pieces of pizza, and 2 apples.
Oh my gosh.
Wow. That sounds like a post-night out.
That's a breakfast of champions. They threw in the apples to be healthy.
Yes,
yeah. Right.
So
the balance is
spent way too much time looking at this table and thought it was great. So at least from this, there were no foods that really seemed to stand out.
So apple now, a day.
Yeah,
maybe
true.
2 apples.
Yeah. So then they switched to a mouse model, and they repeated this experiment in mice who were either fed or fasted, and then fed again, and really saw pretty much the same overall phenomena in the mice. And they saw it both in black 6 and in Balb C mice. Same changes in cytokines, same changes in T cells in vitro. Really a pretty similar phenomenon. They also looked in OT1 RAG2 mice, where they knew all of the cells were naive. And they saw that they could still see improvements in metabolism. So it did seem like the naive cells were getting this metabolic improvement. And so they next kind of wanted to know, all right, what about effector functions of the T cells? Mm-hmm. So they made OT1 cells that were fed or that were fasted, that were different based on a congenic marker, on a CD45 marker. So they could transfer in both fed and fasted cells into mice and compare the responses of those cells.
That's a great experiment.
And so they transferred those into mice and then infected them with Vaccinia ova to look at the responses to Vaccinia ova. And it seemed as though the cells that came from the fed mice were better able to expand than the cells that came from the fasted mice. And that's
antigen induced. That's not just general proliferative
exactly.
proliferation.
This is antigen induced after infection with Vaccinia
So
ova.
When they parked them in, they parked equally well.
They parked equally well. They looked at the difference with expansion. They even switched the CD45.1 and CD45.2 to make sure that it worked. They also saw that the fed cells had higher levels of metabolism, as you might expect. And that was kept over 7 days in culture as well. And so they basically said that it looks like under physiologic conditions, there is this difference in these cells. They also looked at memory cells. And so the fed cells seem to have more memory cells, or make more memory cells than the fasted cells, in terms of both proportion and absolute number. And the memory cells seem to have elevated longevity for 40 weeks after the initial infection. And so one of the things, so then the next experiment they did is they wanted to see whether those memory cells were, as they call them, functionally superior memory cells. And they did that by re-challenging them with a synthetic antigen, which was DEC205 ova, linked to a monoclonal antibody to DEC 205, and they measured the expansion of the cells, and they saw that the fed and the fasted memory cells expanded equally well. So here they said it didn't look like the memory cells were necessarily better. I sort of would wonder if they looked at other models, like an infectious model, what they would see. And there's another model they use later on that makes me think that this might be really model specific.
I have a lot of questions about translatability, but I think I'm going to hold off until the end, because part of me is like, okay, like it makes sense. If you give the T cells more metabolites, glucose, they're going to have greater capacity to do their functions. And then, okay, it might not be that I don't want to take it too far, but like, should I just be eating all the time so that I can better protect myself against influenza? But I imagine there's like, you eat too much, you have maybe...
Yeah.
We can get into translation, but it's just a curious model. I guess I'm wondering like why this model, but I think that you're totally right in that. And this is one thing that it gets into a little bit later that I was thinking about later. I think that, they don't do things like if they have excess calories or something like that. It's just, did you eat, 6 hours before this? It's kind of like eat breakfast though, right?
It's really just eat breakfast.
Yeah.
Because then your T cells will be better ready for your day.
Exactly. In fact, they also did this same experiment and changed the timing between when they did the infection and the transfer. And so it seemed like if they pre-infected the mice or did sort of earlier challenge, the fed T cells had more of an impact. Whereas if they transferred the cells, waited a while, and then tried to challenge, they had less of an impact. So it seems like it's really kind of pretty close after the feeding.
Yeah, and acute.
It seems like it was definitely an acute thing.
Can I ask, did they compare T cells they took from fasted mice with just unfasted mice? Because what I wonder is the conclusion is that feeding causes the T cells to then be better. But could it be actually that fasting changes the metabolism of T cells to make them not quite as good?
So that's interesting. So they don't so basically it's just mice that are given ad lib access to food versus mice that are not given access to food for 12 hours and then given access again.
Okay.
And so it's your question is sort of a little bit of a tricky question. But there's another
would you have wanted to see, rather than ad lib compared to fasted and then fed, looking at fasted versus fasted and fed? I wonder whether there are just compensatory differences that happen when you fast mice, because they're fasted for how long?
So I'm
like 18 hours.
I'm double checking. So fasted for 12 hours.
That's pretty long for a mouse.
Yeah. So let's see. And I'm just double checking that.
We might not have all these answers, but it's interesting to think about.
Yeah, the effects of fasting itself on the cells and how that could change their responsiveness to food.
But also that's very relevant because everybody's doing these fasting windows now, right? So they do this timed eating or whatever.
Well, I think that there is one experiment that does kind of address some of these questions, though it's not exactly the way that you're talking about. So before they got to that experiment, they also did some RNA-seq of the fed and fasted cells.
Of course.
And of course they saw, some differences.
They were different.
But I don't know that there's anything, super big to write home about. But then what they actually went to next is they said, is this difference in the T cells due to some serum metabolite that's impacting the T cells? And so they took serum from the fed or fasted mice and incubated it with different T cells to see if the serum could convey the phenotype. So it's not exactly the experiment you're talking about, but it does.
It still starts to get at mechanism. Yeah.
And so what they found is that when you had fasted T cells and you gave them fed serum, they had better metabolism and they acted well. But if you had fed T cells given fasted serum, there was no impact. And so it seemed as though it was really the having been fed gave you some metabolite that was helping T cells. It wasn't that fasting was harming the T cells. And so that's sort of the best that they could sort of do to address some of those questions. They also checked to see whether there were differences in the microbiome and saw no microbiome differences. And they looked at differences in insulin and insulin receptor and didn't see differences in insulin receptor. So they decided to then try to figure out what the metabolite was. And they looked at the different metabolites in the sera from the fed and the fasted mice. And they specifically looked at a bunch of different metabolites and looked at a whole bunch of different compounds. And they found that specifically certain types of fatty acids were the difference. And so there were different fatty acids in the fed mice's serum that seemed to give the CD8 T cells their ability to function better.
And do you think, or did they comment on whether they thought that was some sort of signaling response, whether there's a receptor, or is it just a presence of a metabolite that can be fed into the metabolic pathway and churned up? So
they kept going and actually figured out specifically, they were looking at specific triglycerides that were particularly important. And they talked about chylomicrons, which was a word I'd never seen before, but they're part of lipid binding to the LDL receptor. And they specifically were the things that were influencing the CD8 T cells. And so they looked at, they did a bunch of things here. And they really saw that it was the chylomicrons. And then in future experiments, they actually showed that, LDL receptor was necessary. And they also looked specifically downstream and saw that the changes in these fed T cells were changes in translation, not in transcription. They had very similar transcripts, big differences in translation. And that those translational differences were happening via mTOR signaling. And so they basically were saying that these triglycerides were signaling specifically to mTOR through the LDL receptor, and that was improving CD8 T cell function.
It makes sense that they would use a rapid adapting process like that, by like translational regulation rather than transcription, which takes just a little bit longer.
Yeah, absolutely. And when they talked about all of the metabolites, this was another place where some of Steph's questions sort of came up, because I know that I've seen talks where people have talked about, high fat diet and this kind of diet and that kind of diet. And I wonder if, too much of these triglycerides would be a particular problem.
Right.
They're just sort of having some in the serum versus not. And so I think that actually trying to figure out the levels could be another really interesting question here. So all of these are really interesting observations. And then they decided to do some experiments that they thought about with translation of this. And these weren't the first things that I would have thought of with translation, but they are pretty interesting. So what they actually did is they said, well, does this mean that we would get different immunological outcomes if we're doing cell therapies? So if we take cells out of a patient, say for CAR T cell therapy or some other kind of therapy, will the T cells act differently if we take them out after you've eaten versus after you have not eaten?
I like that experiment.
No, I like the experiment too. It was just not the first thing I would have, I hadn't thought of it.
It is so funny to think about how this paper came about. Were they interested in this question first and then dove into mechanism and presented it in the opposite order?
Yeah, I have no idea. But so they basically used a model where they had mice, a tumor model. So they had mice with a specific tumor antigen. They expanded T cells took T cells with these. They took mice with these T cells. They were either fed or not fed. Then they took out so they had fed or fasted T cells. They transferred them into other mice with tumors.
And they modified them with a CAR first?
First, these were not modified with a CAR, but they did a CAR as well.
Okay.
So they just did the straight non-modified T cells. Mm-hmm. And they saw that the T cells from fed mice were superior therapy in clearing the tumor. And they also use some human CAR T cells and just looked at their action in vitro and saw that the human CAR T cells were had more effector function in vitro if they were coming from a fed donor as opposed to a fasted donor. They also did another set of experiments with mice, and this is one that I thought was particularly interesting. So they used CAR T cells in a mouse. And what they found, and the thing that was really interesting was they found that the mice who got the fasting T cells had more frequent tumor relapses than the mice that got the fed T cells. So it seemed as though the fed T cells were acting as memory cells or were persisting longer than the fasted T cells. And that does sort of go back to some of the memory data they presented earlier in the paper. They didn't see a difference in response to synthetic antigen, but here it looks like the cells are superior memory cells in preventing tumor relapse in the mouse.
Well, and just in general, the T cells have higher interferon gamma secretion, right?
Yes.
Cytokines.
Yes, they have higher cytokine. Yeah. They have higher interferon gamma secretion.
Right.
Yes. And so they their idea in thinking about translation is whether or not this would be useful for someone who's going to be doing some kind of T cell therapy.
That's so interesting.
Which I think is really cool. My first question was sort of, should I eat breakfast before getting a vaccine?
I was exactly thinking the same thing.
That was exactly what I was thinking about, which they don't show. But they, and they talk
you can make
nicely.
some assumptions, though, based on their data. What we would do.
Yeah, you can make some assumptions. And so they sort of note that the immune system is responsive to sort of immediate, as they call it, immediate changes in systemic metabolism. They point out they didn't control for dietary input. They didn't there's sort of a lot of diverse factors that could go into this, but I just thought this was a really interesting set of experiments, asking a question that I hadn't even necessarily thought to ask, but something that could be useful in cell therapy, something that could be something to take into account in terms of vaccination or some other translational type of thing, and maybe something we should spend some more time thinking about.
One of the things that crossed my mind was a situation where people are often fasted and where there's an increased potential to have an infectious event is surgeries, right?
Yes.
We have to fast for anesthetics, right?
Yep.
But yet at the same time, this could potentially make you more susceptible to
so fascinating
infections.
That's a great point.
And what about colonoscopies too? In the event that there's some perforation or anything. Yeah. I don't know.
But of course,
I would not want to be awake during that.
There's a limit. You can't eat too much.
Well,
right.
Yeah.
So
that's also a problem.
Thinking about like malnutrition and the other way that you would,
Because if they did these experiments with the obese mice, I think they would get a different result.
Yeah.
Right?
I think that with most of the experiments with the obese mice are sort of long term, eating in excess. Mm-hmm. And so it would both be interesting to know whether there's been some sort of shift in those mice after the long term dietary change, but also kind of what happens just if you have one big, meal, and it's an acute type of thing. But I guess this made me think about all of these questions that would be interesting to have answers to, and that are certainly questions my students would be asking me.
Neat paper.
Yeah.
I made sure to eat breakfast this morning.
In preparation for.
Yes, in preparation.
So we should eat three square meals a day, basically.
I think we should. I think what I learn whenever I look at anything about sort of trying to think about this advice is that, like, the things my grandmother told me were probably right.
But we know already that in countries with a lot of malnourishment, measles is worse, for example. Right?
Yes. Yep.
Enteric vaccines don't work as well.
Yeah.
Right. But do we necessarily always know how much that was related to changes in the GI tract versus the immune system?
Right.
Correct.
I don't think we know.
I mean, that's why proper nutrition is part of you can give countries vaccines and everything, but you need to make sure they're fed also.
Yes, absolutely.
Because as you said before, there are places where people can't eat properly. So
I'm a snacker, so I'd like to think that I'm just always maintaining those T cells at some high functional level.
If you look at the table, it is clear that some of the folks are snackers.
Okay.
Because it says the last thing they ate, which was not necessarily a dinner. And you can tell who are the snackers.
I was just really hoping chocolate was going to be a good thing.
Well, I mean, there are definitely people on here who had, there's somebody with candy for breakfast. There's somebody with chocolate mousse for dinner. So.
Oh. I just want to be able to eat a little chocolate here and there.
I think that's acceptable.
Chocolate mousse as a meal. Cool.
All right. Another cool paper coming up from Cindy.
All right. So I am very interested in innate immune receptors, especially right up my alley, toll-like receptors, and we've been digging in a little bit into potential connections between receptors that recognize nucleic acids in the cytoplasm, which might alert to viral infections, as well as the toll-like receptors and the nucleic acid recognizing ones that are in endosomes and how they might connect or communicate. So this particular paper really interested me.
It's like your lab and my lab communicating.
Exactly. So this paper was published in Nature Communications in March, and I'm probably going to butcher the name of this drug, but it's prasugrel inhibits TLR7-driven autoimmunity in systemic lupus erythematosus by acetylating cGAS. So there's a lot of terms in there that we're going to introduce, but the first author is Guo, and the last author, I think there's maybe 3 corresponding authors, Chuang Fu, Chinhua He, and Tao Li. So as you can imagine, most of these are from China. They're from the Nanhu Laboratory at the National Center for Biomedical Analysis in Beijing, the Department of Rheumatology and Immunology in Peking People's Hospital, Beijing. It's a lot of different ones. Department of Rheumatology at Shanghai Jiao Tong University School of Medicine, Institute of Translational Medicine in Hangzhou, School of Basic Medical Sciences in Fudan in Shanghai, and Institute of Pharmacology and Toxicology in Beijing. So it's a bunch of Chinese people who worked on this particular study. And I was interested in it because, again, I am interested in TLRs, especially nucleic acid sensing TLRs, of which there are primarily TLR3, which sees double-stranded RNA, 7 and 8, which see single-stranded RNA, and 9, which sees DNA. And then there are many different cytoplasmic sensors that can see nucleic acids. cGAS is one particular one that gets a lot of attention, and it recognizes DNA. But the disease that we're talking about here is systemic lupus erythematosus, or SLE, or I will refer to it mostly as lupus. So lupus is an autoimmune disease. It's driven by autoantibodies to DNA, RNA, and other autoantigens, mostly associated nucleoproteins, but can be a lot of other antigens. And the disease is actually characterized by deposition of these nucleic acid immune complexes. So the antibodies and the antigen in complex, they form these larger structures, and those structures accumulate in various different locations and cause inflammation and the general sequela of the disease. So for example, if they get deposited in joints, you get an arthritic inflammation. If they get deposited in the skin, it's associated with a very characteristic facial rash that's called a butterfly rash because it goes across the nose and then flares out like wings across the cheeks. If it gets deposited in the kidneys, it causes glomerulonephritis, so really bad inflammation in the kidneys, or really can accumulate anywhere. And depending on where it accumulates, you can get different disease outcomes. It's really hard to diagnose because the symptoms overlap actually with a lot of different diseases. Because if you don't get that characteristic rash, it just kind of looks like any other arthropathy. There is a genetic disposition. So it definitely runs in families, but it's not one particular gene. Now, this will come up because in mouse models, you can do this very clearly. You can knock out one gene and you get a beautiful lupus-like disease. But in humans, we don't have one particular gene that's associated with the disease. There's been a lot of genes that have been implicated in it, but it's probably a complex multifactorial disease. We do know that there are certain things. So we've been talking about, food in the immune system. So there are a lot of environmental things that can influence and cause triggers in lupus. And those are things like sunlight. So when your skin is exposed, you can cause a exacerbation of the skin inflammation, infections, medications. So, but basically, we really don't understand how this disease starts. We know that there's definite sex bias where women are much more likely to get the disease. And like I said, there are multiple animal models, very different animal models. There are animal models where you inject a drug. One is called Pristane. Another is called imiquimod. So you can paint or inject these drugs and it will induce a lupus-like disease. And then there's also some natural versions of the disease, either are models of it where you knock out a particular gene or you have a naturally occurring mutation or you crossbreed two related strains and they develop a similar disease. So we've had a lot of these different models and a lot of different things, but what has been pretty well generally accepted now is that there is a link with these nucleic acid sensing receptors. Exactly what they do, somewhat still muddy, but there's definitely a connection. And particularly for human and TLR7, there have been gene-wide association studies that have identified polymorphisms in TLR7 that are associated with lupus. So probably there's some connection. In the mice back in 2006, there was a Y-linked autoimmune accelerator, so a YAA mouse. And when they tried to map what the mutation was in that mouse that caused them to get this autoimmune disease, lo and behold, it was a multiple duplication of the TLR7 gene on the Y chromosome. So again, overexpress TLR7, now you get disease. And they've actually done that too, where if you transgenically overexpress TLR7, you can also drive disease. And they've done knockouts of 7, and it's complicated, very complicated, whether you knock out 7 or 9 or both or whatever. But generally, if you knock out 7, you knock out the generation of anti-RNA antibodies. So there's definitely a role for TLR7 in generating those autoantibodies. So this is really interesting. But all of those receptors, like I said, are sort of inside the cell, but not in the cytoplasm. So the part that recognizes the nucleic acid is facing what would be external to the cell, but it's in an endosome. So when something comes into the endosome, it will detect it. So why don't we all have autoimmune disease if there's DNA and RNA floating around and there are these receptors? Well, there are other proteins that are really important for chewing up nucleic acids so that they don't trigger these kinds of responses. So you don't have accumulation of nucleic acids. So one of those players is Trex or Trex. So that one is really important. And if you mutate that one, and that will come up in the paper, if you mutate that or you lack that gene, that allows nucleic acids to accumulate. And when they accumulate, they make it into those endosomes and then they trigger the response. And the interesting thing is that these nucleic acid sensing TLRs are expressed on B cells. So what happens is if you have an anti-DNA, anti-RNA B cell, it will bind to nucleic acid. It will pull it in because that's what B cell receptors do. But it will actually trigger then the TLR in the endosome. And then that causes that B cell to be activated and produce lots of its antibodies, which happen to be autoreactive antibodies. And then those autoreactive antibodies bind more of the nucleic acid. They form complexes, and then those get taken up much more effectively. And then they trigger the cycle. So that's how we think that TLRs are involved in this. But the treatments for SLE really are just general immunosuppression, which isn't great, or some immunotherapies to knock out B cells or CARs, because we just talked about CARs, knocking out B cells. But in order to be able to come up with new drugs to try and treat this disease, we really need to better understand what are the signaling pathways and how do they work. So this paper starts to look at a connection between the TLR7, which I just went into, and cytoplasmic sensors of DNA. And the bottom line is that they look for already approved drugs that might interfere with this pathway. And they had some hint of what was going on before because the group has published some modifications that happen to the DNA sensor in the cytoplasm called cGAS. So they were looking for drugs that target that. But the DNA sensor that they're interested in is a cGAS-STING pathway. And there's one other connection that's really interesting, and that is that that pathway is known to induce high levels of interferon and interferon-stimulated genes. And lupus is often referred to as an interferonopathy because there are high levels of interferon and interferon signatures in people with the disease in their peripheral blood cells. So they were like, maybe there's a connection. And my lab has done the same question where maybe there's a connection. Yeah. And the last thing I want to say is that other papers have already been published that have basically ruled out a role for cGAS in these models. But they used a different model. So there is confusion in the literature, and probably it's not all one disease because there might be different genes mutated. And so depending on which gene mutation you might have a different trigger in that disease. So one of the arguments that this authors in this paper make is maybe some of the patients are characterized by this pathway that we're going to talk about. And so they might benefit more from a particular drug that other patients would not.
Yeah. The one thing I would mention also is Cindy mentioned Trex, and Cindy thinks a lot about TLRs. And from my knowledge of literature, when I see Trex, I immediately say, aha, something's going on with cGAS. Because of Trex's important role in breaking down free DNA in the cytoplasm. And so I think that coming from a TLR kind of background, some of this background information says to you, oh, this must be a TLR thing, especially things like imiquimod being a drug that's important here. But as soon as I see Trex, my thought, because I spend a lot of time thinking about cGAS, is, oh, well, cGAS has to be involved if Trex is part of this. Yeah.
So you're exactly right. And I think what they tried to do is like put it all together. Yeah. So what did they actually do? I like this. I like the fact that this paper started out with human lupus patients. So they didn't just start out with a mouse model. They provided data from human patients that bolster their idea that their model, that there's a connection between cGAS and STING and TLRs and lupus is there. So the first question they asked is, Is there anything in a lupus patient that suggests that cGAS might be activated? Now, you need a little bit more information if you're not a cGAS aficionado to understand that when there is DNA in the cytoplasm, which might be a virus or it might be some cell DNA that gets in there, when there is nucleic acid DNA in the cytoplasm, it will bind to cGAS, and cGAS is an enzyme, and it is a synthetase that makes a molecule called cyclic GAMP or cGAMP. Once cGAMP is made, it can diffuse out or it can actually stay in the cytoplasm and bind to the protein STING. So when cGAMP binds to STING, and STING is usually localized in a complex on the endoplasmic reticulum, but when it binds, it causes a larger complex to form. It also brings some kinases together, including TBK1, that then phosphorylate STING and provide a docking site for transcription factors that are really important for inducing a set of genes that includes interferon-stimulated genes. So it's an interferon response factor or an IRF that binds, and then that gets phosphorylated, and then that goes and upregulates the genes. So the cGAMP is really important in that it is the metabolite that is generated by cGAS when it's activated. So if you go look in the plasma of healthy people and lupus patients and you say, is there any cGAMP? If you see it, it's an indication that maybe there is an activation of cGAS happening somewhere. So what they found was that there was higher levels of cGAMP in the blood of lupus patients, but it wasn't just a generalized inflammation because they tested RA patients and they tested dermatomyositis patients, and it was only up in lupus patients. So they did this in a primary cohort, and then they did a secondary cohort to confirm. And basically, it's about 60 lupus patients total, and they really saw a higher level. So this is suggesting that we don't know why or how it's happening, but there seems to be some kind of activation of cGAS in these lupus patients. So since I've already talked about there is potential role for TLR7 as well, they were trying to look to see, okay, so is there actually a correlation between the level of expression of TLR7 and the level of cGAMP that you can measure in the serum? Because what I told you, for example, was you can overexpress TLR7, or the mice that have too many copies of TLR7 end up with disease. So maybe people who have more TLR7 for whatever reason actually generate more of the cGAMP. And I thought the correlation was meh, but it was there. I think it was like about a 0.46 correlation. It's not a super high correlation, but generally I think the argument is like maybe if you have more TLR7, you actually have a little bit more cGAMP. I thought much more convincing was taking cells, stimulating TLR7, and asking, do you actually see cGAMP induced?
How quickly, what's the degradation of serum cGAMP after TLR7 is activated? And maybe just looking at that in serum wouldn't be accurate?
Good question. Do you know on that, Brianne?
I don't know the half-life in serum, but I know that cGAMP traditionally does have a pretty short half-life. There are a bunch of enzymes that break it down, some of which are cell surface enzymes. And so I know when I use it for experiments, sometimes there's a non-degradable version to try to make it last longer. So I think that's a great point. It's probably pretty short-lived, but I don't know the exact timing.
I guess then if you can measure it, it's pretty high.
Yeah.
In a patient.
Right.
Yeah. But like I said, there was this meh correlation, but I think they made me more convinced by taking mouse bone marrow cells, treating them with a TLR7 ligand and measuring cGAMP production. And in that case, they could see, a time-dependent increase of cGAMP produced by these cells, suggesting that TLR7 is inducing cGAMP. And the only way we know that cGAMP is produced is through cGAS. So somehow TLR7 is activating cGAS. Is the idea. So now they move to a mouse model of lupus-like disease. And I'm going to say like disease, and I'm making air quotes because these models mimic some of the outcomes, but they are definitely not traditional lupus. So we got to take that with a grain of salt. But anyway, what they do is they treat mice with imiquimod or IMQ, and they do this 3 times a week for 4 weeks. And what that does is will cause splenomegaly, increase in antinuclear antibodies, increase in anti-double-stranded DNA antibodies, increase in interferon, renal pathology, deposition of IgG and IgM in the glomeruli, and inflammation of the kidney. So those are all lupus-like signs that you would see in a mouse. And this imiquimod is a TLR7 agonist. So we knew that you can induce this disease with TLR7 stimulation, and they can measure all of these outcomes that look like lupus. What's interesting is that they also saw an increase in plasma cGAMP. So again, associated with the disease in this mouse model is this increase in cGAMP. So then you could say, okay, well, maybe there's some weird thing going on. Maybe somehow TLR7 uses some other pathway and cGAMP is induced by some weird mechanism. So you really have to ask, is cGAS involved? So how do they do that? They use knockout mice, of course. So they have cGAS knockout mice, and then they repeat their imiquimod model to try and induce disease. And what they saw was basically a reduction in all of those parameters that I just mentioned, splenomegaly and all those other things, including cGAMP in the serum and immunoglobulin depositions, et cetera, et cetera. So what do they conclude from this is they're saying that somehow imiquimod, which is supposed to activate TLR7 only, and they did some controls there to make sure that if you lack TLR7, you don't get disease, and you lack cGAS, you don't get disease, and inhibitors and things and cell lines and so forth. So it's pretty convincing that imiquimod is activating TLR7, and then TLR7 shoulder shrug by some unknown mechanism, then activates cGAS, and then cGAS has this manifestations of disease. And again, I want to focus on the fact that other groups have shown no role for cGAS. And they did this with a Pristane model. So there's a drug, Pristane. You can inject the mice. They will get the same sequelae. Did it in cGAS knockout, no effect. This group actually repeated that. So they tried the Pristane model, no role for cGAS, which is interesting. So now you've got different models of lupus where some do depend on cGAS and some do not.
Right. So do you know the details of, so I just Googled it, but it doesn't totally help me. Do you know the details of sort of what Pristane is or sort of how Pristane works?
I do not.
Apparently, according to this, it's a terpenoid alkane from shark liver oil.
That doesn't help me a lot to understand.
It doesn't help me a lot at all. But I guess the reason why I ask is that imiquimod, we know acts through TLRs.
Correct. It binds to TLR7.
Right. It binds to TLR7.
We know that pretty clearly.
And so that could be a sort of obvious difference here of what the initial stimulus is if it's a TLR related or a non-TLR related stimulus.
Absolutely.
The differences between murine and human TLR7 induced cGAS activation, what do we know about that? And is that what could contribute to this difference in the animal models?
It could. There are differences. I think there are certain ligands that will maybe activate 7 from one species and not another, but generally they're pretty similar.
Okay.
The intracellular DNA sensors or the cytoplasmic DNA sensors between mice and human do have a fair bit of variation in terms of which ones are present in each species.
Or in what quantities, maybe?
And even if they exist.
Right.
And so that might be a caveat to this, although it doesn't go in a direction that they're really looking at here.
Yeah, sure.
Yeah. All right. So that brings up the really weird conundrum in that, so it sounds like TLR7 is activating cGAS, but the known mechanism of cGAS activation is by detection of DNA in the cytoplasm. So, so what's going on here? Let me see where I am. Okay. So they said, Well, what do we know and what might be going on? Well, stress of cells can cause mitochondria to leak DNA. And that mitochondrial DNA is a known ligand for cGAS. So they said, maybe TLR7 signaling stresses the cells in such a way that the mitochondria leak their DNA. And it's not that out of the question because TLRs, as we were just talking about metabolic activities, TLR stimulation can completely change the metabolic processing inside a cell, especially a macrophage. So it's not out of the question that it might change the function of mitochondria. So what do they do?
This was the first experiment I wanted to see as I was reading this.
Yeah, exactly. So what they did was they just simply measured mitochondrial DNA in the cytoplasm. And what they saw was you add a TLR7 ligand to cells, and lo and behold, you have an increased level of cytoplasmic mitochondrial DNA. So it's plausible. But then it's like, okay, well, how does that actually happen? And what they talk about is that there are these things called megapores that can form on the outer mitochondrial membrane that will then open and allow mitochondrial DNA to leak out. Those megapores are made up of two main proteins called BAC and BAX. And they have an inhibitor to BAC and BAX. So what that does is prevent those megapores from forming. And the hypothesis is that if we prevent the megapores, we prevent leak of mitochondrial DNA, and maybe that will reduce the cGAMP and the interferon production in response to TLRs. That would implicate the mitochondria in this process. And that's what they saw. So they added an inhibitor to BAC and BAX, and what they saw was a reduced amount of cytoplasmic mitochondrial DNA and then a reduced outcome of TLR7 signaling, which is interferon production from the cells. So that's pretty convincing. It connects these things together. Now, they go a step further, and that's why I really like the mechanism in this paper, is the group previously showed that cGAS could be acetylated. So that's a post-translational modification that alters the function of cGAS protein. And what they showed is when cGAS gets acetylated, its function is decreased. So there's less production of cGAMP. So cGAS is inhibited. And they said, okay, well, we know now that cGAS is important for mediating the TLR signaling to produce interferon and to get connected to this downstream disease in this model. So what if we could look for drugs that would acetylate cGAS, thereby inhibiting its function? And maybe that might be a new way to try and treat at least some forms of lupus. So how did they do this? I thought this was pretty clever. They took existing drugs. So they took 3,159 FDA-approved small molecule drugs. So they're already approved. Why is that important? Because now if you find one that works, you don't have to go through all the approval process. You can already start testing it in people because it's already proven to be safe, right? You could just be an off-label or a new clinical trial to test them. So they screened this compound library, and what they did was in silico first, so by computer-based experimentation, to determine which of those drugs are capable of donating in a acetyl group. And based on computational things, might it actually transfer that group? So they narrowed it down to their top candidates. And then they used recombinant cGAS to ask whether when they incubate it with that drug, does the cGAS get acetylated? And they used, I thought this was kind of interesting, aspirin as a positive control. So by the way, aspirin already does this. So maybe in certain lupus patients, if they're taking aspirin for some reason, they're actually already acetylating their cGAS and blocking the pathway, and who knew? But anyway, that's a little aside. But so they start to test these things, and basically what they discovered was a drug that's in the title, prasugrel, that induced acetylation of multiple lysines on cGAS. It was much better than aspirin, which comes back to the fact that maybe, yes, if they were lupus patients were taking aspirin, it might have had an effect. But also, I think they probably had to take a massive amount of aspirin to have an effect, because this prasugrel was like 1,000 times more active. So you could use a much, much, much lower concentration. And they confirmed that the acetylation happened actually in real cells, human cells, with natural cGAS, so not recombinant cGAS. So that all fit nicely. But then that raises the next question. does the drug actually interfere with TLR7 signaling? And could it be used to do anything? So what they do is they treat cells with prasugrel and then stimulate with TLR7 and measure outcomes. So what are the outcomes? Things like cGAS activation, so cGAMP production, IRF3 phosphorylation. I told you, remember, when cGAS is activated, it produces cGAMP. cGAMP binds to STING. STING comes together and IRF3 binds, and IRF3 gets phosphorylated. So IRF3 phosphorylation, interferon production. And what they found was that the prasugrel suppressed all of these outcomes induced through TLR7 signaling. But it didn't have an effect on poly-IC that they transfected into the cytoplasm. So it's not an RNA sensor thing. And interestingly, they also tested TLR1-2 ligands and TLR4 ligands, but they measured IL-6. And I think that's very interesting because everything we've talked about so far with the cGAS-STING IRF is to interferon response genes, and IL-6 is an NF-kappa B signaling pathway. So the fact that IL-6 is not affected, but they didn't measure interferon.
It might not matter.
I don't know. I think the jury's out of whether this whole pathway might still be involved in various different TLRs, not just nucleic acid sensors.
Interesting. So you're saying they didn't, in that experiment, measure interferon?
No.
Gamma.
The interferon, type 1 interferon.
Oh, sorry, type 1 interferon.
Type 1 interferon. So alpha beta.
Alpha beta, yeah.
Yeah. Anyway, that's my little beef with the paper because I think they switched readouts and didn't read the readout that might be actually informative. And I think that's important because if this is a pathway that's downstream of all TLRs and not just nucleic acid TLRs, it changes how we think about treatment, right? Because now you're going to be shutting down LPS response, response to gram-positive bacteria, gram-negative bacteria, and then that's a whole different ballgame versus just shutting down response to autoantigens and nucleic acids. But regardless, they wanted to know, they took their mechanism thing just a step farther because they were super interested in this acetylation of cGAS thing. So they wanted to know exactly what is acetylation of cGAS doing. And they did this before they went into the mouse model to see if the drug actually treats the disease, which we'll get to in a moment. But basically what they show is that if you activate cGAS, it forms these condensates in the cytoplasm, which is really interesting because, I sort of, when I started learning cell biology, thought about signaling as like, you just activate one thing and then it activates the next thing and activates the next thing. And then if you actually step back and think about how big a cell is and how small a protein is and how one protein might find another protein, you're actually talking about somebody on Earth trying to find somebody on like a planet near Alpha Centauri, right? And they need to come together in order to be able to signal the pathway. And that's just not practical. So we actually know that there are these condensates and these signaling complexes that are formed in the cytoplasm that allow proteins to come together and then be able to transmit the signal. Anyway, so cGAS does one of these condensate things.
Yeah. So I usually I think this whole idea of liquid, it's called liquid phase separation.
Yep.
And the way I kind of think about it is that we often imagine the cytoplasm as being kind of homogeneous goo.
Yeah.
But in fact, the sort of specific chemical mediators or chemical constituents of cytoplasm differs in different places. And that might, be, if we say phase change, more solid or liquid is a sort of simple way of doing it. And different proteins will move to different patches. And so it's sort of like thinking about the cytoplasm as having areas or little mini organelles that are not actually having membranes around them. It's just that the cytoplasm is heterogeneous, and different proteins like to flock to different parts of the cytoplasm. And so cGAS is super well understood to go to specific condensates.
That's such a super important concept to remind us.
Yeah.
Because I think I do sometimes disregard that right? In my conception of the of the cytoplasm.
I started reading some of these papers when they started coming out with regard to cGAS and phase separation and started learning about them. And I took them to my colleague who is a biochemist. And I don't know if I've ever seen him get so interested and excited about something because of how much it got into some of the really interesting areas of biochemistry. And it is a fascinating area. And I think there are some cell biologists who end up spending a lot of time thinking about cGAS because it seems to partition among different condensates so well.
Yeah, I like that partition. From what I hear from some virologists here, viruses do this too, because they form little factories and little areas where they condense all of their proteins to be able to synthesize.
The specific sort of biochemistry of how much it may be liquid phase separation in that isn't always well understood, but I think that it probably is something really similar that people just have to figure out the biochemistry of.
Yeah. So they think that they think that these condensates might be really important, and they wanted to look at what is the connection between acetylation. And they thought, well, maybe when cGAS gets acetylated, it doesn't form these condensates as well, and maybe that's how acetylation of cGAS is interfering with function. And they show that that's actually true. So they make a non-acetylatable mutant, and it forms condensates just fine. Wild type can form condensates. But if you treat with this prasugrel, then it interferes with the condensate formation of the wild type version of the protein, but not the non-acetylatable mutant. So if you can't acetylate cGAS, it still forms condensates and it doesn't get disrupted. So it still signals. Gets a little brain gymnastics there. But basically, the drug is acetylating cGAS, and acetylation disrupts the structure formation. So you inhibit signaling, but then it doesn't have any effect if it can't be acetylated. And they do some stuff in cells to show that they form droplets, which is this liquid interface there, and that's also disrupted by the prasugrel. So basically, they're providing a mechanism by which this drug can work. So it can directly cause acetylation and block this condensate formation. So there's a mechanism for how this might actually work in the disease. So first, I'm going to breeze through this, is basically they use the Trex mice, which is the cool model, because we know that is cGAS dependent. If you don't have Trex, you don't chew up your nucleic acids. The nucleic acids accumulate, they make it into the cytoplasm. Black box, shoulder shrug. Maybe you know, I don't know. But anyway, that will trigger cGAS, and then that triggers the disease. So the Trex knockouts get a lupus-like disease, and they show that if they can treat those mice with prasugrel, and that alleviates the disease. So it looks like maybe this is a way to do this. They did a little weird thing with that particular model. If you want to go look at the paper, all they did was measure interferon-stimulated genes in the heart. So it's not really the best analysis and model, but it was just a control to say, can you actually give it to mice safely? It's tolerable. It's not toxic. They can treat them, and then they can reduce the disease in the Trex mice. But the cool thing is can you actually do this in their lupus model? And this is the coup de grace experiment, right? The great experiment. So they do imicamod 3 times a week for 4 weeks to induce the disease. And they do it with or without prasugrel or controlled DMSO. And importantly, they use one other drug. So they do 3 comparison groups. The third comparison group is because I told you that this was an FDA-approved drug, and it's an approved drug because it has a known target called P2Y12 that's somehow involved in clotting. And I didn't look into all the details of that, but if you're interested in clotting, you should go look that up. And they took another drug that also blocks P2Y12, but doesn't bind to cGAS. They didn't show
So basically,
that.
Is it the clotting function that's
correct
important here, or is it the actual function of binding cGAS?
Binding cGAS and acetylating cGAS, right. So they compare, so that drug is clopidogrel, and they compare that to prasugrel and DMSO. And what they found was that the splenomegaly, the antinuclear antibodies, the interferons, the kidney disease is all reduced by prasugrel, but not by the DMSO control or the clotting inhibitor control, which is kind of cool. And they also have another lupus model. So they had the pristine model, they had the imiquimod model. They have one more model. It's an MRL-LPR mouse that also gets autoimmunity, and that's known to be cGAS independent. And prasugrel had no effect on that disease. So it does seem to needs to be a cGAS dependent disease. And the prasugrel is potentially causing acetylation of cGAS in the mouse, which is disrupting its function, which disrupts all of the signaling and propagation of the autoimmunity, and it reduces the disease burden of the lupus-like disease in the mice.
But so wait, the prasugrel worked in pristine treated mice?
It did not.
Okay, so yeah, I was going to say that.
It did not work in the MRL-LPR mouse. It did not work in the pristine mouse.
Okay, perfect.
It only works in the imiquimod mouse or in Trex knockouts, but that's not TLR7 dependent. So then the very last thing they did, because they needed to connect it back to humans, and this was, you can do what you can do, right? So they took PBMC from lupus patients and they treat them with prasugrel a lot more, but they said they did another toxicity level, but it's 50 microgram per mil in vitro versus they were injecting 20 mg per kg in a mouse. Maybe I should do a calculation there to see what that is. But anyway, 50 microgram per mil in vitro, which they also say was non-toxic. They measured the interferon transcriptional profile, and they said that it reduced the interferon transcriptional profile in the PBMCs from lupus patients treated in vitro, which kind of cool. And what they said was it's similar to the JAK inhibitor, which now I've forgotten the name of, something manipulative.
Yes.
I have it in my notes that I forgot to say it. Ruxolitinib is a JAK inhibitor.
Flows right off the tongue.
Yeah.
Ruxolitinib.
Ruxolitinib. And that one, so that's an inhibitor that drops down interferon signatures because it inhibits interferon signaling. So when you get type 1 interferon, it triggers type 1 interferon receptors that drives more type 1 interferon. So they knew that. And what they showed was that this prasugrel reduced the interferon signature similar to that. So it's a strong inhibitor. They used it in cells from 10 different SLE patients. And then they had another 26 patients that with and without prasugrel in vitro, and they saw suppression of the interferon stimulated genes that correlated with the amount of plasma cGAMP. So remember, I told you that lupus patients have higher levels of cGAMP. So if they have very high levels of cGAMP, they have very high suppression of the interferon stimulated genes with prasugrel. If they have low levels of cGAMP, they get a very low level of suppression. So it's suggesting that prasugrel is actually knocking down whatever pathway is producing that high level of cGAMP that causes the interferon stimulated genes, which is, according to the paper and the data that they show, via TLR7 to mitochondrial DNA to activating cGAS to then activating the downstream thing. So overall, what they conclude is that cGAMP could be a biomarker. So you could draw blood, and if a patient has high levels of cGAMP, that could say, well, they probably have cGAS activation, and maybe they would be the right patient population to treat with this prasugrel. Since it's already approved, technically it could be translated very, very quickly into treating patients and would be a novel way to treat lupus, which would be very welcome for new drug treatments.
Yeah.
They also point out that cGAS drives inflammation related to aging.
It does.
It does.
So maybe this drug can help with aging.
Maybe. I don't know, though. You had to be super careful because assuming it's going to cause you to have potential increased bleeding issues, since it also
yeah.
inhibits clotting.
Yep.
Yeah.
Which you wouldn't think would be great for an elderly population.
Not
Right,
particularly
right.
great.
Right.
Yeah, I mean, their platelets drop anyway. So what does the acetylation of cGAS normally do? Do we know?
So it usually is a sort of a inhibitor, a way to sort of turn
Right.
off signaling.
So what conditions would lead to that? Do we
Good
know?
question. I don't know.
I don't know a specific condition, but I mean, it's basically, how we're going to get an end to our interferon production.
Right.
The only things I can think of are sort of artificial cell culture based things.
Yeah,
Okay.
for now. I was trying to see if I could pull up the other paper that they had to see if they had any natural ways. I don't see anything off the top of my
Yeah,
head.
I mean, there's also some questions that are very much debated about cGAS's ability to access the nucleus and nuclear DNA and things like that. And
Yeah, because people have
modification-
seen it in the nucleus, but then
They have, and it looks like certain modifications will stop it from acting in the nucleus, or it may be in certain locations on nuclear DNA. And so that's the other thing that is sort of unclear on this is, are some of these modifications involved in helping to make sure cGAS is not sensing nuclear DNA.
I will say that when I first read the very first papers about cGAS, and I was like, oh, that's cool. You can see virus in the cytoplasm. I'm like, wait, but cells replicate and they break open their nuclear envelope and then the DNA is there in the cytoplasm. Why is cGAS not activated? But there is actually now several papers that say that the cGAS is important in that whole process, in actually preventing response to the nuclear DNA by binding, which is just,
And
very
the
interesting.
levels of cGAS at different parts of the cell cycle vary in terms of how much cGAS you're seeing. But there are lots of papers that show that when the cell is not dividing, and there's DNA damage, there's micronuclei that will be released from the nucleus and will be detected by cGAS.
Wow.
And there's somebody at my institution that studies cancer cells, and that when they squeeze through very small areas, they pop their nucleus and then they reform it.
Yep.
And that can actually trigger. Yes.
That's
it's wild.
crazy.
Yes.
That is crazy.
Yeah. And the fact that we're doing lots of things where we can just basically make the mitochondria spit out their DNA. So it's a really easy way to turn on cGAS.
Oh, wow.
Becomes pretty cool.
That's cool. Thank you. Thank you, Brianne and Cindy.
Yeah.
Pretty cool papers. That's Immune 107. You can find the show notes at microbe.tv/immune. You can send us questions or comments to immune@microbe.tv. And if you enjoy these discussions, I know you do. That's why you're listening. You should
That's why
support
you made
us.
it to the end, right?
Yeah.
Yeah, I know. We would love your support. Microbe.tv/contribute. Cindy Leifer is at Cornell University over on Blue Sky. Cindy Leifer. Thank you, Cindy.
Thank you. Send papers. We want to see new ones. Tell us what you want to hear about.
Yeah, send us your suggestions. Immune@microbe.tv. Steph Langle's at Case Western Reserve University. Stephanie Langle on Blue Sky. Thank you, Steph.
Thank you. And I just like to say I love that episode 107, we're talking about TLR7.
Yeah. All right.
Yeah. Lucky number 7.
Brianne Barker's at Drew University. Bio-Prof Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.